And today we are presenting about the topic transport in plants. And now I will just start with the past year question first. So we look at the first question. Water absorbed by plant roots travels by different pathways from root hairs to the xylem. Figure 6.1 shows this pathway in the roots of ranunculus acris. And this is a figure 6.1. Is it big enough like this?
Yes.
Okay. Then we look at the question. Name cell A and pathway B as shown in figure 6.1. So this question, you all can answer if you want. Can answer in the chat box. Yeah, the first one is the endodermal cell. And for the pathway B, anyone? Yeah, also for the pathway B is the upper-plus pathway. And here is the answer scheme. And before we move on to the next question, I had actually retrieved some diagram from the teacher's PowerPoint. And just let's look at it. This one is the transverse section of a Dicot root. And we can see that this part which I color here, this part is a xylem. And outside the xylem, there's a layer of tissue, which is called the pericycle. And also outside the pericycle, there's another layer of endodermis. So we go back to the diagram. We can see here, this part is a xylem. And outside the xylem, there's a layer of pericycle. And outside the pericycle, there's endodermis. And one thing to take note is that endodermis is a name for the tissue. And for the question, it asks for the cell, so the cell is called the endodermal cell. And also, let's look at this diagram here. This one is the upper-plus and the sym-plus pathway. The upper-plus pathway is a movement from cell wall to cell wall. And also something that we need to take note here is that the movement across the lignin and the cellulose is also the upper-plus pathway. This is because two of these is not living cells. It's not living cells, so there are no cytoplasm for the sym-plus pathway.
Wait. Can the water pass through lignin? I think the water pass through the cellulose part, right?
Pass through the cellulose
part of the cell wall. Because lignin is not permeable to water, so water cannot pass through the lignin, I think.
This one I'll check later and then I post in the Telegram. Okay, sorry everyone. And for the sym-plus pathway, it's a movement from the cytoplasm of one cell to the cytoplasm of another cell. And here is my way of remembering is that cytoplasm there's a S inside and sym-plus also S inside. So sym-plus pathway is from cytoplasm to cytoplasm. And something to take note for the sym-plus pathway is the Casparian strip and the suberin here. And what is the purpose for these two is to force the water to pass through the cell by sym-plus pathway only instead of upper-plus. So what is the purpose for this? So first one is to regulate the movement. Of water and solute into the xylem vessel. And also the second one is to prevent toxic substances and pathogens from entering the xylem tissue, the xylem vessel. So, okay. So we move on to the next question. Transpiration occurring at the leaves is mainly responsible for movement of water across the root of our acris. Explain how transpiration is responsible for the movement of water across the roots as shown in figure 6.1. So the question here is asking for example, inside a plant, how can the transpiration help the water to move from the roots to the xylem and up the xylem? So for this question, we can write about what happened during the transpiration. So what happened is, firstly, the water will evaporate out of the basophil cells. And second is, the water vapor will diffuse out into the atmosphere through the stomata. And when after the water escaped from the leaf, the water potential inside the leaf decreased and is lower than the water potential inside the roots. And this created tension, or we call the negative pressure, or we also call the suction force. And this led to the transpirational pool. And what the transpirational pool does is, it will lift the water column up the xylem vessel. And we can also say about the cohesion-tension theory here. So we can write about cohesion. Cohesion is the forming of hydrogen bond between the water molecule and water molecule. So we can write cohesion between water molecules. Water molecules prevents water column from breaking. And we can also write about the adhesion here. Adhesion is the forming of hydrogen bond between water molecule and the cellulose wall. So we can write adhesion between water molecule and cellulose helps the water column to climb up the xylem vessel. Okay, so by this we answer the question, and here is the mark scheme. Okay, so we move on to the next one. Explain why the movement of water in path C is slower than in path B. So this is a diagram exactly from the question. So as you can see, the pathway B is here, is from the cell wall to the cell wall. So the pathway B is apoplast pathway. And pathway C is the dotted arrows from cytoplasm to cytoplasm. So it's the simplest pathway. So in this question, we should explain why simplest pathway is slower than apoplast pathway. So something that we can write here is the movement of how is the movement of the simplest pathway. So the water in the simplest pathway, the water passes through the cell membrane and the tonoplast. And the water move from the cytoplasm to another cytoplasm through the plasmodesmata. And why is it slower is because it has greater resistance. And here is a mark schema. Greater resistance, and water need to pass across the membrane and through the plasmodesmata, so it's slower. So next question. Ions are taken up by root hair cells using active transport and facilitated diffusion. Describe two ways in which facilitated diffusion differs from active transport. So in this question, we just need to talk about the facilitated diffusion. How is it slower? So there's a diagram here. This part is a facilitated diffusion, and this is an active transport. So how is facilitated diffusion slower? Anyone want to answer? Yeah, Andrea got it. For facilitated diffusion, there is no ATP involved. And another one, anyone want to try? Okay, if no one, oh, okay, Zhixian got it. For the facilitated diffusion, the ions move down the concentration gradient. Because as we can see here, for the active transport is against the concentration gradient, and for passive transport is moving from high to the low concentration. Here is low to the high one. Okay, so here is the answer scheme, and we move on to the next question. So Miriam Olinder is a xerophytic plant. A photomicrograph of a section through the leaf of an Olinder is shown in figure 3.1. So the question is, the leaf shown in figure 3.1 has a number of adaptations to reduce water loss by transpiration. Two of these adaptations are a multi-layer epidermis. So we can see here, there are multi-layer epidermis, more than one layer, and also stomata only found in depressions known as stomata crypts on the lower surface of the leaf. So this is a stomata crypts. And so the question asks, explain how a multi-layer epidermis and stomata crypts will help to reduce water loss in an Olinder. So for the multi-layer epidermis, what we can write here is, it increases the distance for diffusion. It increases the distance for diffusion. Hence, it reduces the rate of transpiration across the cuticle. And for the stomata crypts, what we can write here is, what the stomata crypts do is it will trap the moist air in the atmosphere. And as it traps the moist air, it reduces the water potential gradient between the stomata air spaces and the atmosphere. And this also reduce the rate of transpiration through the stomata.
Johan,
The
second point, is it substomatal air spaces? Substomatal air spaces?
Is it substomatal like this? Okay. This one I will check later also. Okay. And one thing that I do here is I actually write across the cuticle and through the stomata here. I don't know is it needed, but because there are three types of transpiration, which is through the stomata, across the cuticles, and out from the lenticles. So I'm just mentioning which type of transpiration happening here. Okay. Then here is a marking scheme. So we move on to the next one. Sucrose, amino acids, and other assimilates synthesized in palisade mesophyll cells of an Olinder pass to the vein, where they can be transported within specialized cells from the source to the sink. Name the cells specialized for the transport of assimilates in an Olinder. This is a quite straightforward question. Anyone want to try?
I think, Johan, you call the name better because only 2 persons will answer so far.
Okay, then I'll call Jing Er.
Is it the CPU?
sieve tube is the tissue name, and here is the cell.
Oh, companion?
Sorry?
Is it the companion cell?
No, it's not the companion cells. The companion cell is the cell beside the sieve tube. So you are half correct. It's through the sieve tube, but sieve tube is the name for the tissue, and the question is asking for the cell. So the
Wow,
cell.
so it's sieve tube element.
Yeah, you got it. Sieve tube element. Okay, thank you, Zinger. Then we move on to the next question. Explain the difference between a source and a sink. So I'll just answer here. What is the source is? The source is the site of synthesizing. And the sink is the site of receiving. And what for it receive the substances is for growth or for storage. An example that it can be given here is the example for the source is the palisade mesophyll cells, which synthesize the sucrose amino acids and the assimilates. So you can write here the palisade mesophyll cells. And the example for sink, there are storage compound like the fruits or the roots. Okay, so here's the marking scheme, and we move on to the next one. Figure 4.1 shows the movement of sucrose from source to sink through the phloem in the plants. So this is the figure 4.1. Okay, then we see the question. With reference to figure 4.1, name an example of a source and a sink. Also, anyone want to try this question? Example of source and sink. Sink tubers, yes. Zhixian got it. Tubers. And for the source, yes, the leaf. And in the masking, there are more examples there. So after this, you all can look at the examples. And the next question, name cell C and D. So cell C and D. Anyone want to answer? There are points. Yeah, Andrea got it. C is the sieve tube elements and D is the companion cells. So I no need to write. Okay. Then we see the next one. With reference to figure
Hang
4.
on. I want to see the answer for 4A part 1, the source. Can you read everything about the source for the mark scheme?
The source is leaf, mesophyll, palisade, spongy. And the mesophyll, palisade, and spongy is actually all inside the leaf. And why is it? Why the leaf is a source is because the leaf carry out photosynthesis and all the substances needed are created there. And the sink is that fruits, the flower, the fruit seeds, and other else, which they cannot produce the substances that are needed by themselves. So they need to receive from the leaf where it is produced.
Okay, thanks.
Then we move on to the next one. With reference to figure 4.1, explain how sucrose travels from the source to cell C. So we go back to the diagram. From the source to the cell C. And inside this diagram, there's actually an error that I have been made sure with teacher already. Here is here. There shouldn't be anything blocking the plasmodesmata, and this core transporter should be here. Should be here. And just cancel this from your mind. Nothing is here. It's empty spaces.
Yeah, I think the co-transporter should be in the membrane. So this diagram is a bit strange. Huh?
So, what's the question again? how the sucrose move from the source to the sieve tube element. So we can see here, the sucrose is back here, and these are the sucrose. And here, this one is the companion cells. This one is the companion cells. And in the companion cells, there's actually something called the proton pumper, which will pump the proton out. And beside this, there's also the proton sucrose co-transporter, which will help the sucrose to enter the companion cells by facilitated diffusion. And after the sucrose enter the companion cells, the concentration of sucrose here has increased. And when the concentration increase, the sucrose will diffuse into the sieve tube elements down the concentration gradient. Okay. So the sucrose is here already. So we answer the question. And let's look at the mark scheme. Okay, the mark scheme say the proton pump out and the sucrose enter through the co-transporter. And the role of companion cells here is having the proton pump and the proton sucrose co-transporter. And the sucrose diffuse down the concentration gradient from the companion cells into the sieve tube element through the plasmodesmata. Right, so we see the next one, from cell C to the sink. Cell C to the sink. So inside the cell C, inside the sieve tube element, when the sucrose enters here, it decreases the water potential inside the sieve tube elements, and hence the water from the neighboring cells and the xylem vessels will enter the sieve tube elements by osmosis down the water potential gradient. And water enters here, so the hydrostatic pressure here will increase. So like this, here the hydrostatic pressure here will be higher, and so the hydrostatic pressure will push the sucrose down. Will push the sucrose down the sieve tube, down its pressure gradient. So sucrose will be transported until it reaches the sink, and it will diffuse into the companion cells again, like this one. So we look at the mark scheme. So, the water potential decrease, so the water from the xylem vessels will enter, from the xylem vessel and neighboring cell will enter the sieve tube element by osmosis down the water potential gradient, and hence the hydrostatic pressure has been built up, and it moved down the pressure gradient, and until it reached the sink, the sucrose will be unloaded. Okay, and this also called the mass flow, which is when a large quantity of substances are moving in one direction only. So we see the last question, I think. Explain why multicellular plants require transport system for substances such as water and sucrose. So here, we look at the mark scheme. So here we can say that what problem does multicellular plants facing? The problem is it has small surface area available for diffusion compared to its volume. So we can say the distance for the diffusion is too far. Hence the diffusion rate is very slow. And the site when the cells are requiring the substances, are too far away from the site where it is produced. So we need a transport system to overcome the limitation of diffusion. Okay, so the idea of mass and bulk transport. Mass and bulk transport is also the same with with mass flow. When the large quantity of same substances are being moved in one direction only. So here we answer the question. So that's all for me, and I will pass it to Ying. Okay. So,
Yeah, so good morning, everyone. Well, now I would like to invite everyone to enter the Kahoot for a small quizzes, or can be said as a quick revision. Please enter the link by the link, and then enter the pin to participate. Thank you.